In this screencast, we are going to talk about the biological effects of radiation as well as half-lives and different ways to measure radiation. So when we talk about the biological effects of radiation, it differs based on the type of ionizing radiation there is. When we discuss this, it's important to, let me define shielding first. So when we discuss this, we're going to talk about shielding, which is a way that we can protect ourselves from radiation. And so when we talk about shielding, this is just protection from harmful radiation. And so one of the things you can see here is we've got alpha radiation. It travels a few centimeters through the air, and then you can see tissue penetration. It's going to stop at the skin surface. It's really only dangerous if it's inhaled or eaten. And you will see this in pop culture pretty frequently in kind of like, those procedurals like NCIS or 24, where somebody inhales or eats alpha particles, and that's very detrimental. For protective shieldings, paper and clothing will shield you from alpha particles. For beta particles, they travel a few meters. That means if you're out, if you're past a few meters, beta particles aren't there anymore. So they will not penetrate past the skin layer. And so heavy clothing, plastic, aluminum foil, gloves will protect from beta particles. X-rays will go several meters and they'll penetrate tissues but not the bones. That's why they're used to image like bone breaks in your teeth. And so a lead apron, like if you go to the dentist, you wear a lead apron, or a concrete barrier will prevent X-rays from going through your body. Now, gamma radiation can travel several hundred meters. It will fully penetrate the body, and thick lead, concrete, or a layer of water will protect against that. Now, when we talk about biological effects of radiation, it's important to understand that biological molecules are damaged when they encounter ionizing radiation. And when that happens, this causes changes to cellular processes. And so we talked on the previous slide about the different penetration levels, but I love this image because it shows a skin layer, it shows muscle, bone, and organs, and you can see where alpha particles stop at the skin. Beta particles can travel through a little bit farther to the muscle, and then gamma particles go all the way through. X-rays, of course, would be stopped right here. So when we discuss ionizing radiation, we talked about this in a previous screencast, right? When high energy radiation, like these gamma rays, pass through a cell, they collide with molecules, causing them to lose one or more electrons, and that's why they're so dangerous. So let's see, high energy radiation. I just want to give you some words for that. Like gamma particles pass through a cell, right, and collide with molecules, causing them to lose an electron, right? And it can be one or more. Now, tumor cells are more susceptible to the effects of gamma radiation than normal cells because they're really dividing frequently. So gamma radiation has been used to treat and cure cancer, but paradoxically, it can also cause cancer in healthy cells. So we have to be really careful when we use these cancer treatments, right, as a society. Now, we can produce radioactive isotopes. So we have naturally occurring radioactive isotopes and some that we can produce. And these can be prepared by bombarding stable isotopes with fast moving alpha particles, protons, or neutrons. And so I have an example here of a neutron being bombarded. So when it's being bombarded, you're going to see it in the reactant side. And so basically neutrons are shot at this molybdenum nuclide, and we produce a radioisotope or a radioactive isotope this way. So it's a common way to produce these radioactive isotopes. So we can find radioactive isotopes naturally, or we can make them in the lab. Now we have to have units to help us with the measure of radiation or activity. And so two of the units that we use, and there's lots of different units. So two of the units that we use are a becquerel and a curie. Now I also have a millicurie here and a microcurie, and I just want to remind you from kind of those first lectures, milli means there's 1,000 in one, and the micro means there's 1,000,000 in one. So those are back to those metric prefixes. Now, with the Becquerel and the Curie, and these were named for Henry Becquerel, who kind of discovered radiation, and Marie Curie, who also investigated radiation. These are independent of the nature of radiation and the effect on biological material. Right? So those are helpful, but they don't tell us anything about the nature of the radiation or the effect on biological material, because as we've talked about already, gamma rays react differently with biological material than alpha particles do. So another unit that's used is a Röntgen, and this is spelled okay, and it's pronounced like rent and then gen, right, just like that. And this measures high energy ionizing radiation. Okay, and it measures radiation, like the interaction of radiation with air. Okay, so it measures the interaction of radiation with air. And that means there's no real info about the biological effect on the tissue, okay, because we're all dealing with the air here. So there's another unit, it's a rad, and it stands for the radiation absorbed dose, right? And that takes into account the absorbing material. A similar unit to this is a gray, which is abbreviated as a GY. And that's also that measured absorbed dose. So you'll see both of those. But one of the ones the next two I feel like are the most helpful for us, or the next one is the most helpful for us. It's a REM. And I have seen two different abbreviations. The abbreviation that your book uses, which is what we're going to stay with here, is the Röntgen equivalent for man. right? Which describes the biological damage. Caused by absorption of different kinds of radiation by the human body. Okay. And so these are the different units that you're going to see when you work problems, trying to figure out how much radiation is left after a certain amount of time. And so it's helpful to know what the differences here are. Now, there's another term. It's not a radioactive unit. So not a unit. Let's start there. But I want to talk about it before we go on to the next slide. And that's the lethal dose, which a lot of times is referred to as the LD50. And you run into this with radiation, but you can run into this with any chemical. And it's just the dose that would be fatal to 50% of the population. No matter where you go and work after college, you are probably going to encounter some sort of chemical. And your employer has to have a SDS or a safety data sheet on file for whatever chemical you encounter. And the lethal dose for 50% of the population, whether it be inhaled or ingested, is a lot of times shown on the SDS sheet. So this is something that we run into in industry all the time. And so I just wanted to mention it here. It's also used in radiation. Now, one of the things that we can use to measure the amount of radiation is a Geiger counter. That's shown here. Right? And so this would be what you would hear like clicking in movies. You know, if you see like, if you watched any movies, you kind of hear a click, 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 click sound. That is a lot of times what you used. What's used more often now are badges. And so you can see here the radiation strip. The amount of radiation you're exposed to in an amount of time matters too. And so these badges are what people wear if they're working with radioactive elements or, say, at a nuclear power plant. So the other thing that we can use is half-life. Half-life is T1/2. That's the symbol for it. And that's the time required for 1/2 of a given quantity of radioactive isotope to undergo change. Okay, so not all radioisotopes decay at the same rate, and that rate just means speed. And so isotopes with very short half-lives are going to decay very quickly, and they're going to be very unstable. Now, that doesn't mean if you have a long half-life, it's less dangerous. It's sometimes quite the opposite. But short half-lives, unstable decay. Long half-lives decay much slower. So I want us to look at some of the half-lives. And so to put some words to what I just said, natural occurring radioisotopes generally have long half-lives, right? Radioisotopes are generally used in medicine, have short half-lives. We don't want that stuff hanging around very long, right? And so some examples here. Tritium has a half-life of 12.3 years. Carbon-14, that's used for carbon dating, has a half-life of 5,730 years. Radium-226 has 1,600 years. Uranium-238, that's used to date the age of the Earth, has a half-life of 4.5 billion years. Well, these radioisotopes here are used to in therapeutics or medicine. And you can see the half-lives that are listed here. Some kind of really interesting examples here. You've got chromium-51. That's a lot of times used for things like blood volume, to determine your blood volume. Iron is used for the blood as well. Technetium-99 is used in a bunch of stuff, but it's also used in like bone studies. Generally, it's complexed with a phosphate compound because you have phosphate in your bones. It's also used for some brain studies. Iodine-131 is used in the thyroid, right? Thallium-201 is used in artery diseases. It's not listed there. And it's interesting with these half-lives because, for example, I had a friend who had a problem with her thyroid. And so they sent her iodine-131, and she had very specific rules. So they lived her and her husband lived in a one-bedroom house. And so there for a while, I think for a month, she couldn't be around any babies because if you think about it, babies have rapidly dividing cells, so you don't want any radiation to affect their growth. And then for the first 8 days, she couldn't use the same bathroom as her husband or sleep in the same room. So he slept on the couch. And I don't remember what they did for bathrooms. I think he had to go visit a friend's bathroom. I mean, it was really wild. And then my father has had a lot of heart problems. And when Evan was born, he had been exposed to iron-59. And so we were kind of concerned about that. But it turned out to be fine. But that was just kind of always a concern. So let's talk about some problems you'll solve with half-lives. So when you have a question where it asks you to calculate the amount of an isotope remaining, the first thing you want to do is, one, determine the half-life, right? Or the number of half-lives that have passed, right? And then two, you want to determine the isotopes remaining, right? And so we do this by taking 1/2 and raising it to n, which is the number of half-lives, and multiply it by the starting material. And that will tell us the number of isotopes remaining. So I want to do an example of this. Okay. We have radioactive iodine-131. It has a half-life of 8 days. If a dose with an activity of 200 microcuries is given to a patient today, about how much radioactivity will still remain after 32 days? And then we've got a half-life of 8 days. So I've underlined the important things. And so the first thing we want to do is figure out how many half-lives have passed. So we take 32 days, and we know that one half-life is 8 days. And that equals 4 half-lives. Okay, so 4 half-lives have passed. So the next thing we're going to do is figure out how much still remains, because that's what's asked. And so we're going to take 1/2, and we're going to raise that to the 4th. Now, when you do this math, you should get 1/16. Then you're going to multiply that by the amount there, which is 200. All right, and you should get 12.5 microcuries, and it's 12.5 microcuries no matter which way we slice it. Now, there are multiple ways to solve a problem. The half-life is not always going to work out for you in a nice whole number. So you may get like 4.2 half-lives have passed, and you can still use this equation to figure out however many half-lives have passed. But if you have a nice whole number, the other thing you can do is you can say, look, you can rationalize through it. I start with 200 microcuries. I know that when a half-life passes, half of the material gets turned into something else. So now I'm only going to have 100 microcuries left. So that's one half-life. And then I'm going to go through another half-life, and 50 microcuries are going to be left. And then another half-life, and we divide by 2, and 25 microcuries are left. And then the fourth half-life, we're going to have 12.5 microcuries. This is a lot more complicated if it's 4.2, for example. But you can solve this both ways and see that. Now, another common question that you can be asked instead of how much is left is how much decayed. And then what you have to do is you have to take the starting amount and subtract the active amount, and that will tell you how much decayed, right? So if I was asked how much decayed here, I would take 200 and subtract by 12.5, right? Microcuries. Let me write microcuries there so we're not missing any of our units. And we get 187.5 microcuries, right? And so we have 12.5 that are left, 187.5 microcuries have decayed. So let's talk about different applications that we see nuclear chemistry being used in. The first one is a positron emission tomography, or a PET scan. And in this, what you have is a compound is labeled with a positron emitter, and it's injected into a patient. Right? And so different functions can be studied here. Blood flow, oxygen, glucose metabolism, they can all be studied. And so when we look here, right, the person is put into a machine, and there's these radioactive isotopes. They emit a positron, and you have counters, kind of like a Geiger counter here, that can detect the gamma rays, right? And so the gamma rays are produced when the positrons and electrons collide. And so we've got this detection, and so you can build these really nice images. Labeled glucose a lot of times is used to study the brain, and I find this image to be fascinating. Here's a normal brain, here's a brain with mild cognitive impairment, and then here's a brain with Alzheimer's disease. So 3 of my 4 grandparents had Alzheimer's. And so I just find this to be really fascinating as the images of the brains change. And as a matter of fact, it can get even more complex than this because one of my grandparents had Alzheimer's, but then the other 2, one they didn't exactly know what type, but the other 2 had a different type of dementia. And so their behaviors were really different. So anyway, I find that to be fascinating. Other medical applications for radioisotopes. This is an example of a thyroid, right? And so we talked about iodine 131 being used in the thyroid. Okay. And you can see kind of a healthy thyroid here. You can see a thyroid with a cold spot here. And so this is that imaging done with iodine 131. Now, it's important to expose patients to the smallest possible dose of radiation for the shortest period of time. And so, as I've already mentioned, radioisotopes with short half-lives are selected for use in nuclear medicine, and iodine 131 has a fairly short half-life of 8 days. Now, so the 2 main uses of medical radioisotopes, just to summarize, are diagnosing diseased states and 2, therapeutically treating those diseased traits. Now, if you have a radioisotope used in therapeutics, right, that's called a tracer. So if you hear somebody talking about a tracer, that's what it's called. So we've talked a lot about that diagnosing disease states, right? So the PET scan, and you could see diagnosing a diseased state with a thyroid. But now I want to talk about this therapeutically treating. So radioisotopes can be used as cancer treatments in external beam radiation therapy. Gamma radiation is generated from cobalt-60, right? And it's aimed, is aimed at a tumor, and that destroys the tumor tissues, right? Now, there's brachytherapy. It's another way that cancer is treated, right? You have small titanium seeds, right? And those contain radioisotopes. Are implanted in a tumor. And so here is an example of those titanium seeds. My uncle had prostate cancer several years ago, and they implanted the titanium seeds around his prostate. If you are a fan of Grey's Anatomy, these were used in some sort of brain surgery thing. And it was, really dramatic because the person was supposed to be wearing gloves and they couldn't do it and all of that business. So this is one of the ways, or these are two of the ways that radioisotopes are used to treat cancer. Now, we're going to end with another application, which is nuclear energy. And so nuclear energy is uses the process of nuclear fission. And in nuclear fission, we have heavier nuclei split into lighter nuclei. And this releases a large amount of energy, right? Its use, of course, is in nuclear power plants. Right, and the atomic bomb. Right, and so what happens is you have uranium-235. It's bombarded with a neutron. It starts a chain reaction, and in this chain reaction, you form other radioisotopes here, here, and here, and then you form more neutrons that can go on and bombard more uranium. I love this image, right, because it's showing the reaction here. I want to draw your attention to the coefficient shown here, because you may say, hey, Dr. Pierce, when I add up 137, 97, and 1, it doesn't equal 236 on this side. Well, with the coefficients, you have to take 2 and multiply by 1. You'd have to take 3 and multiply by 1 here, right? So just kind of be aware of that. The other thing I wanted to talk about, so we talked about fission. I want to talk about nuclear fusion. Right? And this is a combination of two nuclei forming a larger nucleus. So for example, if you have 2,1 hydrogen plus 3,1 hydrogen, that goes to 4,2 helium, and it forms a neutron, and it forms energy, right? And so an example of where this occurs is the sun, okay? So helium are combining there. I made a mistake here. I put in a dash. I need to not do that. 3, 1. There you go. So this ends our discussion of nuclear chemistry. If you have any questions, please feel free to stop by my office.